Microbiology Chapter 7.5 Using Biochemistry to Identify Microorganisms. Accurate identification of bacterial isolates is crucial in a clinical microbiology laboratory because the results often guide treatment decisions that directly impact patient outcomes. For instance, accurate identification of the causative agent in food poisoning cases is necessary for physicians to prescribe the correct treatment. Similarly, during a disease outbreak, identifying the causative pathogen is vital for implementing effective strategies to control the epidemic. There are various methods to detect, characterize, and identify microorganisms. Some techniques depend on phenotypic biochemical characteristics, while others utilize genotypic identification. The biochemical traits of a bacterium provide valuable information for classification and identification. Analyzing the nutritional and metabolic capabilities of a bacterial isolate is a common method for determining its genus and species. Key metabolic pathways that bacteria use for survival will be covered in microbial metabolism. This section will explore several methods that employ biochemical characteristics to identify microorganisms. In microbiology, traditional identification methods primarily rely on cultivation processes that utilize various media to enumerate, isolate, and identify specific microorganisms. These methods have been used for many years and continue to play a crucial role in laboratory routines, especially for rapid identification of pathogen in medical diagnostics. While traditional methods are cost-effective and provide valuable quantitative key and qualitative information about microbial diversity, they are labor-intensive and time-consuming. Key steps such as media preparation, dilution, plating, incubation, counting, isolation, and characterization can take several days to yield results, and false positives are common due to similarities among microbial species. Moreover, culture-based methods are unable to identify non-culturable cells. Phenotypic identification method, another traditional approach, often rely on chemical reactions to detect microbial growth. For example, a common visual method involves observing increased turbidity in presence of a substrate, with results compared to a control test using a Wickerham card. However, this method can be challenging to interpret and typically requires overnight incubation. Immunoassays like enzyme-linked immunosorbent assay or ELISA are more efficient but come with higher cost and limited applicability to certain bacterial species. The Analytical Profile Index API model integrates these standard methods into miniaturized reaction couples, generating analytical profiles that can identify nearly 100 taxa. Though regarded as the gold standard in clinical microbiology until 1992, this method is still time-consuming. The 1990s saw an emergence of automated identification method that aimed to streamline and reduce the time required for analysis. Variations of the API system such as BBL Crystal and Vitex system automate bacterial identification and antimicrobial susceptibility testing AST from patient samples. The Vitex system employs a broth microdilution technique monitored by an optical scanner to detect bacterial growth within 18 to 24 hours and yeast growth within 36 hours. While these systems offer versatility, some microorganisms may yield unreliable results. The Biolog Omnilog system also provides a rapid assessment methods for identifying bacteria based on their oxidation of various carbon sources, comparing results to a database through a simple yes/no reactions. The students can Google these automated identification methods for their own curiosity, but they are beyond the scope of this chapter section. As advancement in biochemistry and instrumentation have occurred, the reliance on traditional methods has decreased. Modern biochemical methods provide significant advantages, including shorter analysis times and ability to identify multiple microorganisms simultaneously while maintaining accuracy. Certain microorganisms possess specific compounds stored as granules within their cytoplasm, aiding in identification. For example, polybeta-hydroxybutyrate is a carbon and energy storage compound found in some non-fluorescent bacteria of the genus Pseudomonas. Species within the group can be classified based on the presence or absence of the PHB and fluorescent pigments, with examples such as human pathogen Pseudomonas aeruginosa and the plant pathogen Pseudomonas syringae, both of which do not accumulate PHB granules. We have already discussed the visual identification of microorganisms by microscopy. Although microscopy can elucidate many structural identification cues, including some biochemical properties using differential staining technique, they are very limited. In addition, we have discussed molecular techniques such as PCR, RT-PCR, real-time PCR, RFLP, PFGE, ribotyping, etc. Molecular techniques are fast and effective, but may need more time and some specific reagents to perform certain analysis. On the other hand, modern biochemical and biophysical methods leverage the physical properties of the microorganisms for identification. Mass spectrometry being the most popular, as it does not require any sample strain-specific reagents. To identify a strain of Staphylococcus aureus is no different than analyzing a virulent strain of E. coli. Unlike PCR, mass spectrometry does not require organism-specific reagents, and that's why the process is fast and can be done in minutes. We will discuss some of these state-of-the-art techniques in the following slides. The slide compares microbial identification methods over the years with cost and time to get results. Research in microorganism identification has advanced primarily by focusing on reducing the time needed for identification of specific microbes in routine diagnostics. The introduction of semi-automatic and automatic systems based on biochemical methods has been a significant breakthrough in this field. Mass spectrometry is definitely the fastest, but one may argue about the initial cost of instrumentation, which can range from $100K to well over $1 million. But these facilities are not required in every clinical microbiology laboratory. Usually a regional medical clinical facility, federal, state, or mostly private, have these facilities, and all the regional clinical labs send their samples to them by overnight FedEx to get a result in 24 hours. Currently, a method is considered successful if it provides results within 24 hours, with even quicker turnaround time being critical in emergencies. Mass spectrometry methods have become popular for microbial typing due to their speed, lower cost, simplicity, and applicability to a variety of microorganisms, including bacteria, archaea, and fungi. Advances in MS techniques, coupled with new data analysis, processing, and visualization tools, have enhanced our understanding of biological systems by allowing the analysis of various biomolecules such as proteins, lipids, carbohydrates, and amino acids. Additionally, several ionization and separation techniques can be integrated with MS, including gas chromatography, matrix-assisted laser desorption ionization time-of-flight mode, MALDI-TOF, electromigration techniques, and electrospray ionization or ESI. The above photographs are from the Clemson University Mass Spectrometry Facility located in the Hunter Hall. Liquid chromatography mass spectrometry LCMS and liquid chromatography tandem mass spectrometry LCMS-MS are powerful analytical techniques used primarily for separating and analyzing compound in complex mixtures. Here's an overview of how each method works. First is liquid chromatography. The separation is the first step. The process begins with liquid chromatography, where a sample mixture is injected into a chromatographic column packed with a stationary phase, often a solid material. A mobile phase, a liquid solvent, is passed through the column. Retention. The different compounds of the mixture interact with the stationary phase to varying degrees, causing them to elute or exit the column at different times. This separation is based on factors like polarity, size, and charge. Second is the mass spectrometry, or MS, ionization. After separation, the eluted compounds enter the mass spectrometer where they are ionized. Common ionization techniques include electrospray ionization or ESI and atmospheric pressure chemical ionization or APCI. Ionization converts the molecules into charged particles or ions. Mass analysis. The ions are then directed into a mass analyzer which measures their mass to charge ratio. This analysis provides information about the molecular weight and structure of the compounds. Third is the tandem mass spectrometry. Second stage of ionization. In LC-MS-MS, after the initial mass analysis, selected ions can be further fragmented in a second mass spectrometer. This process is called the collision-induced dissociation or CID, where ions collide with an inert gas and break into smaller fragments. Second mass analysis. The fragments are then analyzed again to provide additional structure information. This two-stage analysis allows for greater specificity and sensitivity in detecting compounds, making it particularly useful for complex mixture. The applications of LC-MS or LC-MS-MS are widely used in various fields, including pharmaceuticals, environmental analysis, food safety, and clinical diagnostics. They are particularly valuable for quantifying trace levels of compound in samples and identifying unknown substances. The slide illustrates a typical LC-MS chromatogram spectral analysis. The x-axis shows the mass-to-charge ratio of every molecular fragment, while the y-axis shows the retention time of each molecule in purple. The z-axis is the intensity of the signals. Higher the intensity peaks represent higher amount of the molecular fragment or ions. When analyzing chromatograms, the first step involves identifying distinct peaks corresponding to individual compounds. Each peak associated with a retention time, which aids in identifying the compounds based on known standards. In the mass spectrum, the molecular ion peak typically represents the intact molecule, while fragment ions revealed structural insights. By integrating information from both the chromatograms and mass spectrum, researchers can quantify amounts. By integrating information from both the chromatogram and mass spectrum, researchers can quantify compounds accurately and identify unknown substances by comparing them to a vast database using powerful computation and bioinformatics by an automated process. MALDI-TOF MS is a cutting-edge tool currently utilized for the quick identification and classification of microorganisms by analyzing the mass spectrum of a specimen and comparing it to a database containing known mass spectra for thousands of microorganisms. This technique is based on matrix-assisted laser desorption ionization time-of-flight mass spectrometry and employs disposable MALDI plates, where the microorganisms is mixed with a specialized matrix reagent. The sample and reagent mixture is exposed to a high-intensity pulsed ultraviolet laser, which causes the ejection of gaseous ions produced from the microorganism's various chemical components. These gaseous ions are collected and accelerated through the mass spectrometer, with their velocity determined by their mass-to-charge ratio, resulting in different arrival times at the detector. A plot of the detector signal versus mass-to-charge ratio produces a mass spectrum that is uniquely correlated to the organism's biochemical composition. By comparing this mass spectrum to a library of reference spectra from known microorganisms, it becomes possible to identify the unknown microbe. An alternative to MALDI-TOF, which may present issues related to the use of chemical matrix mixed with the sample, the laser utilization of desorption and ionization of the analyte, is a technique known as electrospray ionization. This method analyzes sample in a liquid state with ionization occurring at atmospheric pressure, eliminating the need of the lasers used in MALDI-TOF MS. Because of this unique feature, ESI-MS has a wide range of applications for microbial identification. Gas chromatography coupled with mass spectrometry (GCMS) is widely utilized for identifying complex biological mixtures due to its effective separation efficiency and ability to deliver distinct mass spectra for diverse metabolites. The GC system comprises a gas supply injector and column, allowing for data analysis through constant flow or pressure. GCMS is particularly effective for nonpolar molecules such as lipids and is applied in microorganism classification based on lipid components. While it provides sensitivity and accessibility to commercial libraries, a major limitation is its requirement for analytes to be volatile, necessitating the time-consuming derivatization for non-volatile metabolites. To enhance performance, technologies such as GC-GC time of flight (MS) can be integrated, offering broader metabolite detection and improved sensitivity, though at a higher cost. Microbes can also be identified by measuring their distinct lipid profiles. Fatty acid of lipids can differ in chain length, presence or absence of double bonds, and quantity of double bonds, hydroxyl groups, branches, and rings. To identify a microbe by its lipid makeup, the fatty acid found in their membranes are analyzed. A widely used biochemical analysis for this purpose is a technique employed in clinical, public health, and food laboratories. This technique detects unique differences in fatty acids and is known as fatty acid methyl ester analysis. In a FAME analysis, fatty acids are extracted from membranes of microorganisms, chemically modified to create volatile methyl esters, and examined using gas chromatography. The resulting GC chromatogram is compared with reference chromatograms in a database that contains data for thousands of bacterial isolates to identify the unknown microorganism. Thank you.